So to begin, I just want to start where we left off. This is the organization of the nervous system as we understand it. The nervous system is divided into two major sections. One, which we've studied already, has been the central nervous system. So when we did the sheep dissection here, we talked about the brain and the spinal cord. We're going to go ahead now and move on over here to the other part of the nervous system, which is the peripheral nervous system. And that's all the nerves and neurons outside the brain and the spinal cord. They're divided into two major sections: motor nerves. Those are nerves that go out and do things. So a nerve that sends a signal out to my muscle would be a motor nerve. And nerves that come in, bringing information, are sensory. So if I touch my finger to something hot, that information comes back into my peripheral nerves via a sensor neuron. Let's just look over here at the motor neurons. Those can then be further divided into those which we have control over, somatic. Somatic means body. These are things I can have voluntary control over. Or the autonomic. That's just a fancy way of saying involuntary, things I cannot control. And we've already found out that the involuntary nerves can be both sympathetic or parasympathetic. And I'll just go to the next slide here and remind you that sympathetic nerves, right, were those which do fight or flight reactions, and parasympathetic, in general, do feed or breed. So if I have a meal and my digestive system is rolling, that's probably something I don't have control over, an autonomic function. It's probably a parasympathetic because it's a feed or breed. Then we went ahead and did a dissection of the brain. Now, we did sheep brains. We didn't do human brains, but just to let you know, this would be a dissection kind of the male brain, and we found that different parts of the brain, of course, have different functions. We have different parts like that are large, and then parts which are very small, like doing household chores there, can be way found in the back of the brain and barely noticeable. And of course, there's the female brain, which also is very different from the male brain and has parts which are very large and parts which are very, very small. I'll just leave this for what it is. And then we went ahead and studied the neuron. And the neuron is the basic nerve cell, and that tells us how information gets out of the brain and spinal cord through the peripheral system to the rest of the body. And information is gathered by dendrites, and it can be either positive or negative information. And all that information then is coordinated or integrated into the soma, the cell body. And if it decides there's enough information, then it generates a nerve impulse of electrical activity, which goes down this long tail called the axon. And it comes here to the synapse, or what we call the terminal cleft here. And this is where neurotransmitters are produced, and we'll talk about those another time when we get into a discussion about synapses, about how this information will go to various things like muscles, which will actually do the work. Now, after this review, what I really want to talk about today, though, is how electrical activity goes down the axon here. In what form? So how is it that a neuron is a specialized cell which can generate electrical activity? Well, it happens with ions. So let's first of all just draw a little picture of a cell. Now, I know that nerve cell has an interesting shape. I'm just going to keep it easy and, let's just say that's a nerve cell. Okay? Now, the nerve cell is actually covered with a couple of different ions. So those ions are going to be sodium, potassium, and phosphate. So let's draw them in here. That's a little too thick. Let's do a thinner one. There we go. Now, most of the sodium is actually found outside of the cell. And as you recall, sodium has a positive charge. Most of the potassium is actually found inside the cell, and it also has a positive charge. And there's also chloride, and most of the chloride, let's try black here, is found inside the cell, and chloride has a negative. Okay, so remember, this is inside the cell, or intracellular, and this is outside the cell. Very good. Now, these guys are charged ions, right? Now, if I go ahead and take a voltmeter, which measures the difference in charge, let's hook one up. This is what it would look like. So let's get a good color here. Oh, how about this one? Good. So I'm going to go ahead and put a probe inside the cell, right? And I'm going to put a probe outside the cell like this, right? And then I'm going to take these wires and hook them all up together. And I'm going to have a little voltmeter on here that's going to tell me how much electricity there is. Okay. The amount of electricity, the voltage, equals the ions on the inside divided by the ion charge on the outside. Sorry about using this weird color. I probably shouldn't have chosen that one there. Let's switch on over before it gets too weird. Okay. It turns out that there are more negative charges inside, especially because of the chloride, than there are outside. So the inside relative to the outside is going to be negative. So this dial here would tilt on over to the negative side. Fair enough? The inside relative to the outside is negative. Very good. We call this the resting potential. And the resting potential of the cell is about negative 70 millivolts. Okay? Now, why is it that all this sodium doesn't come in and all this potassium doesn't go out and all this chloride doesn't go out? And the reason is, again, the neuron has a semi-permeable membrane, and so things can only enter in and out at various times when the cell wants them to. So when it's resting, this sodium can't get in here. And when it's resting, this potassium can't get out. Okay? So far, so good? Let me say it again. Sodium on the outside, potassium and chloride on the inside. More negative charges on the inside. So the inside is negative relative to the outside. How much more negative? About negative 70 millivolts. Potential voltage is all about the differences between two things. Now, when the soma decides to generate electrical activity, it's going to do that. Because if you look, let's see if we can get to the next screen here. If you look at this cell membrane, there are actually channels or gates in there. Remember we talked about channel proteins being inside the bilayer membrane. Well, some of these channels, like this one over here, only let potassium go through it. And some of these channels over here only let sodium go through it, right? So, what would happen if I opened up this one here? Well, since most of the sodium, remember, was outside, if I open this up, sodium would then enter in here. Now, sodium is positive, right? So that's going to change the charge. See, we're starting down here, right? What happens if I opened up this guy over here? Well, if I opened up this way, since most of the potassium was inside, it would then go outside of the cell. Once again, changing the charge of the cell inside relative to outside. Okay, so we start out with sodium on the outside, potassium on the inside, and we start out with a charge difference of negative 70. Here's what happens when the soma decides to change things. Let's see if we can get our steps over here. Okay, we're going to get a number of things that happen here. And all these things together are going to be called the action potential. And an action potential is a way of saying electrical activity is generated, creating a nerve impulse. Okay, remember we're starting here at negative 70. Why don't you go ahead and just take a moment and draw this down, and then I'll go ahead and tell you what each of these steps looks like. So I'll pause for a second and let you draw this down. Okay, let's begin again. Now let's go to the steps, and I'm going to come back to this in a moment, but I want you to write down the steps. Here we go. A stimuli arrives which changes, so the soma has decided now to generate a nerve impulse, right? It changes the membrane potential higher than the threshold. Aha! Let's take a look at that picture again. So we get a little bit of activity, and let's do something, like here. We get a little bit of activity here, and we get enough of a change of the potential so it goes from -70 to -55. That's the threshold. Now, once that threshold is reached, everything breaks loose, and we're going to have huge changes. So let's look at the first big change that occurs. The first big change that occurs is going to be that the sodium gates, remember I showed you there were channels in there? The sodium gates are going to open up. Once that happens, sodium rushes in from the outside to the inside, going down its concentration gradient. Now, what happens to the voltage of the cell? If a bunch of positive sodium rushes in, what happens to the voltage of the cell on the inside? It becomes a lot more positive. We call this increase in positive charge depolarization. Depolarization because we're changing the polarity of where the ions are. And let's take a look at that in the picture that you drew a moment ago. Here we go. As positives rush in, right? Here's the sodium rushing in. It jumps way up here to about positive 40, and we call this depolarization. See how the voltage goes up? More positive. And it goes all the way up here, and when it gets way up to the top, the cell decides to do something else. And the next thing it decides to do is open up those gates, right, that have potassium. And once it opened up cell gates, since most of the potassium was inside the cell, the potassium rushes inside the cell to the outside. Now the potassium was also positive, so if the cell is now losing all its positive potassium ions, what happens to the voltage of the cell? It becomes more negative. We call that, in other words, a return to where it used to be. We call that repolarization. Let's take a look here at what that looks like. So, let's pick out a different color. Repolarization. Here we go. We're now losing all our potassium from the cell. It's going out. And then we get repolarization. Since it's losing the positive, it gets more negative. In fact, it gets so negative, so much potassium leaves that it overshoots its resting potential where it started at, and it goes way down here to about, maybe -80. Wow. Well, that's no good. We call that a refractory period. Now, why does it do that? Well, the reason it does that is because when it's way down there, that stops another action potential by being generated. And the reason the cell wants to do that is to make sure that the nerve impulse only moves in one direction, unidirectional. We want it to go down the axon to the end. We don't want it to go back up, like a wave oscillating back and forth, back up to where the soma was. Okay, so that's the refractory period, right? But you still have a problem. You're way down here and you want to get back to where you started from. So how are we going to do that? And the answer is, to get things back to where we started from, we need a little help. And the help we're going to need now is we need to have pumps kick in. So the gates close, and then we have a sodium-potassium pump which kicks in, which is now going to restore the sodium and potassium concentrations to where they are. Remember, at the end of repolarization, all the sodium is now inside the cell, and all the potassium is now outside the cell. That's not where they should be. They're in the wrong places. So if the sodium is inside the cell, how are we going to get it outside? How are you going to get it to move against its concentration gradient? The only way you can do that is have pumps kick in to pump it using ATP back to where it began. So let's go to the last part of our picture here. Last part is, how do we get back up here? Let's use this color over here. How are we going to go back up from refractory period to back to the resting state? You need to have pumps kick in active transport, right, using ATP to pump against the concentration gradient to get those ions back to where they belong. And then the system is all reloaded, and another action potential can be generated again by the soma.